Now let's take a look at some of the immunologic methods for diagnosing microbial infections. So the antibodies that we form during immune reactions are not only important in combating infections, but they also give us insight into a patient's history of contact with microorganisms. The characteristics of the antibodies that we see in our patients can reveal the history of their contact. And the underlying basis for all of this is serological testing. Serology is the branch of immunology that traditionally deals with in vitro, as in the test tube or in the glass dish, diagnostic testing of the serum, as opposed to in vivo, which would be in the body. Antibodies have extreme specificity to antigens, and they fit like gloves. They are powerful tools for identifying and quantifying antibodies, or for a matter of fact, antigens. We can either use a known antigen to detect an unknown antibody, or we can use antibodies of known identity to detect an unknown antigen. So here we have a patient's serum with the antibody content unknown. If we expose it to a prepared or known antigen, there may or may not be a reaction. In this case, we're seeing agglutination, in which the antigens that we know are being held together by antibodies in this unknown serum collection. So this colony contains an unidentified antigen. When we mix it with the antibodies we know, we can tell that that particular antigen is there because of this molecular reaction where we see clumping of these microbes with the antibodies. So again, this illustrates how we can use a known antigen to detect an unknown antibody, or we can use antibodies to detect an unknown antigen. Before we look at some of the specifics, let's take a look at some of the general features of immune testing. There are many diverse immunologic tests available, using antibodies and antigens as tools. After we look over this quick overview, we'll take a look at each individual type. There are several different types of tests: agglutination, precipitation, complement fixation, fluorescent antibody tests, and immunoassay tests. The most effective tests have a high degree of specificity as well as strong sensitivity. By specificity, we mean that there are no false positives. That is, we're focused only on a certain antigen or antibody. In this case, we're looking at the antigen, the pink plus sign, and its particular antibody. This other antibody is not picking up that antigen. Now, sensitivity means that we're going to detect it in every positive person. That is because very small amounts of antibody are picked up by the antigen. So ideally, we're both specific and sensitive. How do we visualize antigens and antibody interactions? Well, if we have agglutination or precipitation, we can actually see it with the naked eye. But some of the smaller antigen-antibody complexes will require special indicators like fluorescence dyes or radioactive isotopes in order to visualize the quantities present. So the primary basis of serology testing is the binding of an antibody to a specific molecular site on an antigen. In this case, the endpoint reaction is visible to the naked eye. When we consider agglutination versus precipitation, we're really just talking about a difference of whether whole cells are clumping around antibodies or there are complexes in free solution of antigen fragments and antibodies. So here you can see agglutination. We have whole cells with their epitopes, and the antibody is binding to each of the epitopes and holding the cells in a clump. Precipitation also involves clumping. However, we have antigens that are not whole cells floating around and being clumped by the antibody. They behave a little bit differently. Here's an example of how we might see these antigen-antibody interactions. The test on the right here is the tube agglutination test. Each of these individual tubes has a dilution of the patient's serum. So 1 to 20, 1 to 40, we dilute it further and further and further. And then we add the same amount of antigen to each tube. So we're trying to determine what the patient's antibody level is, what their titer is. And we measure their titer by finding the highest dilution of serum that produces a visible reaction. So in that case, we have this tube. So 1 over 160 is showing us the highest dilution of serum that's producing some agglutination or precipitation. Here they're agglutinated cells because agglutinated cells will literally fall to the bottom when the tubes are centrifuged. So again, agglutination versus precipitation. These ones we can see with our naked eyes. The essential difference between agglutination and precipitation are the size, solubility, and location of the antigens. Agglutination is where we have the antigen-antibody reaction producing visible clumps. For example, when we do ABO blood typing or RH factor typing. Also the rapid plasma reagent test that's used for syphilis. Those are seen on the cards over here. You can actually see clumping of the cells in this particular area. The Weyl-Felix reaction is a similar one that's used to diagnose rickettsial infections. These agglutination tests these days have largely been replaced by fluorescent or genetic methods. However, cards are still used in developing countries where the newer technologies are not yet available. Precipitation reactions happen in a test tube. This is where the antigen is a soluble molecule that's made insoluble by the antibody. In this case, you have an antiserum placed over a particular antigen solution, a very gentle placement. And at the border, where the antiserum sits on top of the antigen solution, there might be an opaque zone if the reaction is positive. One classic example of a precipitation test is the venereal disease research laboratory test. Another technique is the Western blot. It's used for detecting proteins and is a close relative of the Southern blot, which is used for DNA identification. The general idea, though, is that we pass a current across a gel. You may have done DNA gel electrophoresis at some point, but maybe you haven't. But imagine that this is a gel in a dish. We pass current across this gel in the solution that it's laying in. At the beginning of the gel, there'll be some wells in which we put certain protein fragments. And as we pass current across the gel, from negative to positive, the fragments will pass through the gel in this direction. Those fragments that are smallest move fastest, and those fragments that are largest get less far. This makes sense because it takes them more time to move through the thick gel. I know this might be a little bit abstract. After we have the separation of the fragments, you can't actually see them in the gel. So in order to get an image like this, it has to be transferred to special blotter paper that binds each of these protein fragments in place. That's why we call it a blot. And then we incubate this blot with a solution of antigen or antibody, depending on what it is we're looking for, labeled with a radioactive fluorescent or luminescent label so that we can see it. That's how we end up with this luminescent purple visual here. Essentially, we're measuring how big particular antigen-antibody complexes are as they move through the gel. Based on their size, we can determine what sort of antibody or antigen these substances are. Another test is the complement fixation test. In complement fixation, we're testing for antibodies to a certain infectious agent. Some antibodies require a complement to complete lysis of their antigenic target cell. These particular antibodies are called lysins or cytolysins. When the lysins and the complement system come together, they act on red blood cells and hemolysis occurs. And lysis-mediated hemolysis is called complement fixation. In this image below, we can see what would happen. If the person has this antibody in their system, then it binds to the antigen. Because the antibody is bound to the antigen, the complement fixes. When we put in sheep red blood cells with lysins on their surface, in this case, because the antibodies have bound the complement proteins, there is no lysis of the cells, and thus we have an opaque solution. If the person does not have this antibody in their system, then there's nothing to bind to the antigen, and thus the complement proteins don't bind to that antigen. And then when we're exposed to the red blood cells with the lysins, the complements are free and they fix to the lysins on the red blood cells and cause hemolysis of the red blood cell, which causes them to leak out all of their hemoglobin. All the particles fall to the bottom and we have a clear pink solution. Let's take a look at a quick animation to help clarify complement fixation.
The complement fixation test is used to test for the presence of specific antibodies in a patient's serum. Patients differ in the amount of complement in their serum. To standardize the complement fixation test, the first step is to heat the serum to destroy the patient's complement. A measured amount of complement and antigen are then added to the serum. In patient A's serum, the complement is fixed by reaction with the antigen-antibody complexes. In patient B's serum, there are no antibodies to complex with antigen and the complement remains free. To determine whether complement has been fixed, sheep red blood cells and antibodies against sheep red blood cells are added. In patient A's serum, the complement is already attached to the antigen-antibody complexes in the serum and cannot induce sheep red blood cell lysis. Consequently, the red blood cells settle to the bottom of the tube. In patient B's serum, the complement is free to react with sheep red blood cells' antibody complexes, resulting in lysis of the sheep red blood cells. The serum turns a pink color as the red blood cells lyse.
Let's have a concept check to see if that made sense. If you performed a complement fixation test and the red blood cells lyse, what's your conclusion? Does the patient's serum have antibodies to the pathogen, or does the patient's serum not have antibodies to the pathogen? Well, if we take a quick look back here we see if the patient has no antibodies, then we get lysis of the cells and thus a pink solution. If they do have antibodies to the pathogen, then there's no lysis of the cell and thus an opaque solution. So the cells lyse, thus B is the correct answer to this question. Another tool for visualization of antibodies and antigens is using fluorescent antibodies and immunofluorescent testing. The fundamental tool in immunofluorescence testing is a fluorescent antibody, which is made from monoclonal antibody labeled with a fluorescent dye. Fabs, or fluorescent antibodies, can be used in a couple of different ways. First of all, with direct testing, and second, with indirect testing. In direct testing, we have an unknown antigen, usually a cell or a tissue, and then an antibody that's labeled with a specific fluorescent dye. When they're incubated together and then using fluorescent microscopy, you can then see cells that are susceptible to that particular antibody under that microscope. So direct testing uses an unknown test specimen or antigen fixed to a slide, and then it's exposed to fluorescence antibody, which is of known composition. In indirect testing, the fluorescent antibodies are antibodies made to react with the Fc region of another antibody. So in this case, we have a known antigen, and then we put a couple of different antibodies in the serum. Antibody 1 is in this serum, and it's going to attach to this particular cell. Then fluorescent antibody is added, and those fluorescent antibodies stick themselves to the bottom of the Y, and they glow. If there's no antibody in the serum, the fluorescent antibodies cannot bind to the cell, and thus there's no fluorescence shown. This image below shows a great image of that fluorescence microscopy. Immunoassays are another series of techniques that we can use to label things. Microbiologists and immunologists have great tools that are being used increasingly in athletics, in criminology, in government, as well as in business to test for trace amounts of substances such as hormones and metabolites and drugs. Traditional techniques in serology are not refined enough to detect very slight quantities of these molecules or these chemicals. So we can use radioisotope labels, we can use enzyme labels, or really sensitive electronic sensors. Radioimmunoassay is a hard one to understand, but we're using radioactive isotopes to label antigens or antibodies. We use these isotopes to pinpoint minute amount of corresponding antigen or antibody, so either part could be labeled. We can compare then the amount of radioactivity present in a sample before and after incubation with a known labeled antigen or antibody. And essentially, at the end of it all, the more of the bound radioactive component there is after the incubation, the less of the unknown test substance there was in the sample. Now, I know this is a tough one to understand, and I couldn't find a good animation to help you out. So don't worry too much about the details, except for this part about utilizing the radioactive isotope. Finally, we'll look at ELISA, enzyme-linked immunosorbent assay. This is where we use an enzyme and an antibody complex instead of an antibody and a radioactive isotope. Essentially, we form an antibody complex that can be used as a color tracer for an antigen-antibody reaction. Let's look at a quick animation of how ELISA works, because it'll be much more productive than me explaining the figure on the right.
The enzyme-linked immunosorbent assay, or ELISA, is an extremely sensitive test that is used to detect antibodies or specific antigens. The direct ELISA is a test for the presence of antigen. In this procedure, a known antibody is adsorbed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained. The colorless substrate for the enzyme is then added. Development of color indicates the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well, and the antigen attaches to the walls of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added, followed by addition of the colorless substrate. Development of color indicates the presence of the antibody being identified.
So all of these techniques that we've been learning about can be used for all sorts of microbes. However, viruses sometimes present special difficulties because they're not cells and they're more labor-intensive to culture in the laboratory. So it's a great example to show the diversity of techniques that might be used to diagnose any sort of infection, but this one's specifically about a viral infection. A Western blot could be used. Electron microscopy could be used. We could look at some metabolic effects. We can examine signs and symptoms. We could use ELISA. We could use a number of different culturing techniques or genetic analysis. So in short, we'd use any number of these different methods to figure out what the infectious agent was. That brings us to the end of our chapter and the end of the lecture component of our course. Thank you so much for all of your wonderful contributions over the semester, and I wish you luck in your future endeavors.